Today we are starting with the introduction to cancer biology. Here is normal liver tissue. Here is our gallbladder. We have right lobe, we have left lobe. This is how a normal liver should look like. Now, this is a liver having metastatic lung tumors residing as the white protrusions shown in this diagram. If we look at it histologically, see these darkly stained cells, these are the tumor cells, whereas these light pink in color, which are the normal cells. How do we define cancer? The response is always rapid, uncontrolled cell division. But is it the right definition? The similar phenomena happens during injury, during wound repair. The cells like fibroblasts, endothelial cells will proliferate vigorously, producing new connective tissue so that the blood vessels can be healed, the wound can be healed. Should we classify it as cancer? So what is actually cancer? The definition, which states that it is uncontrolled cell division, is not wrong, but this definition is incomplete. To define cancer, we look into three different aspects. The first aspect is there has to have multiple mutations. And these multiple mutations, we can say that there is change in the genome, there is change in the gene expression. These multiple mutations will lead to imbalance between the cell proliferation and apoptosis, which is cell death. So, there are multiple mutations which are creating imbalance between cell proliferation and apoptosis. Now, we have to look at the effects: abnormal cell proliferation, abnormal growth, invasiveness, and most important, metastasis. That's how you define cancer. So, cancer is a set of diseases which are recognized by multiple mutations leading to imbalance in cell proliferation and apoptosis, which will ultimately lead to abnormal cell proliferation, growth, invasiveness, and metastasis. And it requires multiple mutations. Now, there is another concept that, for example, this cell right here, which is darkly stained, has undergone a certain kind of mutation and it starts dividing abnormally. One mutation is not enough to make this cell cancerous. There may be development of tumor, but to develop cancer, it requires multiple mutations. And as the cell divides, there are the chances that more and more mutations will accumulate, and as the progression of mutations takes place, the probability of neighboring cells getting mutated becomes higher. And another point is, if only one cell which is mutated is giving rise to tumor, we call it a monoclonal tumor. And if more than one cells, for example, this cell has acquired some mutations, some other cell has acquired another set of mutations, both become malignant and both gave rise to a tumor. A tumor which is originated from different cell lines, we call such tumors polyclonal tumors. So, the point is, the multiple mutations are required for the development of cancer. Now, let's see what are the various effects of these mutations. These mutations will lead to abnormal cell proliferation because they target the cell cycle itself. There is decrease in the rate of apoptosis, there is significant increase in survival rate. The cells which are supposed to die, the cells who have completed their life cycle, they are supposed to die. But when the mutations accumulate, those cells tend to survive. Those cells tend to inhibit the apoptotic pathway. There is withdrawal of contact from surrounding environment. These are the certain mutated cells. They start detaching from the surrounding tissue. They start evading the surrounding tissue. They start degrading the basement membrane and they start invading the neighboring tissues, and eventually they may reach some other organ, some other site as well. So there is withdrawal of contact from surrounding environment. And how is this contact withdrawn? There is degradation of cell to cell interactions. There is degradation of proteins like cadherins. There is degradation of proteins which maintain contact to extracellular matrix. There is breakdown of basement membrane, as we saw in that diagram. Now, these mutated cells will start releasing some lytic factors, some proteolytic factors, which will start degrading the proteins of this basement membrane. The factors such as metalloproteases, which will start degrading this basement membrane so that these cells can cross this basement membrane and invade the surrounding tissues. And ultimately, through blood vessels or lymphatic supply, they can invade some distant organs as well to form secondary tumors. These mutated cells have higher order secretion of metalloproteases, collagenases, and several other lytic factors as well. There is dedifferentiation. As we know, normal cells are well differentiated. If it is epithelial cells, like we saw, these are the cells of epithelium. They are well differentiated. They perform a specific function. But when these cells have to undergo mutations, for example, this cell has undergone some kind of mutation, successive mutations, this cell will undergo de-differentiation. Look at the morphology. Compare its morphology to the neighboring tissue. These cells are epithelial, columnar epithelial, let's say, but this cell has acquired abnormal shape. It is becoming some oval shape or some irregular shape. The cytoplasm morphology will change, the nuclear morphology will change. This is what we call de-differentiation. This cell will no longer perform the similar functions. Yes, it may attain some similar markers, but there is significant change in morphology, there is significant change in function, and this is what we call epithelial mesenchymal transition, or we can simply say de-differentiation. And this de-differentiation will allow these to invade even further, and there is ultimately invasiveness. Invasiveness starts with the local tissue and it will spread to distant sites as well. We can see here, these are the effects of these mutations. There is abnormal cell proliferation, there is decrease of apoptosis, which means increase of survival, withdrawal of contact inhibition. By contact inhibition, we mean normally cells cease to divide when they come in contact with the neighboring tissues. But this property is lost in case of cancerous tissue. There is breakdown of basement membrane by metalloproteases and collagenases. There is de-differentiation. Now, this de-differentiation has started even before the breakdown of basement membrane, but it has not been completed till the very later stages. These cells can proliferate, they can invade during the process of de-differentiation, and ultimately there is invasiveness. If we take these different cells in the cultures, we take normal cells in the absence of serum growth factors, we take normal cells in the presence of serum growth factors, and we do the same with the cancerous cells. Normal cells in absence of serum growth factors cease to divide very early. They reach plateau phase very shortly. Whereas these cells in the presence of serum growth factors will also attain the plateau phase, but they tend to divide until the various factors are being consumed, and there is certain limit which is reached. There is contact inhibition present in these cells, so these cells will have to stop dividing. But in case of cancerous cells, you can see this graph, they do not stop dividing in absence of growth factors, which leads us to the conclusion they become independent of these growth factors. So what happens if we give cancerous cells these serum growth factors? Then they divide indefinitely, but the rate of division will increase significantly. Let's compare the benign tumors with malignant tumors. As we know, every benign tumor cannot become malignant tumor, and it is the malignant tumor which is classified as cancer. I will be comparing normal cell with benign growth or benign tumor, and benign growth or benign tumor with malignant tumor. So if we talk about division in normal cells, these are extremely dependent on mitogens, extremely dependent on the conditions, whether they have to divide. In case of benign growth, they divide, but division is relatively slower. In case of malignant tumors, they divide rapidly, they divide vigorously. Talking about genomic stability, in case of normal cells, genome is well stable, well regulated. In case of benign tumor, genome is comparatively stable, but not as stable as the normal cells. But if we compare it to malignant tumor, it is very much stable, and it has acquired some mutations, but not enough to make it cancerous. As in case of malignant growth, there is genomic instability because multiple progressive mutations led to mutation of certain genes, which has led to progression of the cancer, and there may be some chromosomal abnormalities seen as well. Dependence on the growth factors, or we can say dependence on the mitogens. Normal cells are highly dependent on the mitogens. In case of benign tumor, they may be dependent on the mitogens, they may be losing this dependence on the mitogens, but they haven't completely lost this dependent on the mitogens. Whereas malignant tumors have completely lost their dependence on these mitogens. Let's talk about localization. Normal cells are well localized. Benign tumors are comparatively localized, but they may start invading local tissues. And once that invasion happens, they start proliferation, they start going to neighboring tissues. That is what distant invasion look like. That is the characteristic of malignant growth or malignant tumors. So if we are talking about this invasion, let's look at this diagram. We have normal epithelium here, which is attached to the basement membrane or basal lamina. Here is capillary. For example, some cells has undergone mutations. There is rapid cell division. These cells have started dedifferentiation because they are not looking like epithelium anymore. And when this happens, these cells will release some metalloproteases, some collagenases, some proteolytic factors, by which this basement membrane, this basal lamina, will be broken down. And finally, these cells will start invading the local tissues. After this local invasion, there comes a stage when these cells will start invading the blood vessels, or start invading through blood vessels. We call this stage intravasation. And these cells will start traveling through the capillaries, blood vessels. Now, during their journey, only a fewer cells will survive, and some of the surviving cells will start adhering with blood vessels at some distant sites. And ultimately comes a stage when they will exit these blood vessels. This stage is what we call extravasation. And when they exit these blood vessels, they escape into the surrounding tissue and they start forming the secondary tumors there. This is what we call as full-blown metastasis. So metastasis is a property of malignant growth. Talking about EMT, epithelial mesenchymal transition, there is the beginning of epithelial mesenchymal transition in the benign stage, but there is partial or complete EMT if the malignancy is observed. In normal cells, cell-to-cell contact is well preserved, and in benign growth, cell-to-cell contact is again well preserved. But in case of malignant growth, there is weakened or complete loss of cell-to-cell contact. The similar thing happens with cell to extracellular matrix contact. Let's talk about metabolism. In normal cells, there is well-regulated metabolism. In the case of benign growth, there is relatively normal metabolism. In the case of malignant growth, there is increased metabolism because cells has to divide, cell has to increase its replication, its transcription, its translation. So there is increased metabolism and increased utilization of glucose, which we term as this Warburg effect. So what about pyruvate requirement? In case of benign tumors, there is normal pyruvate requirement, whereas in case of malignant tumor, due to increased metabolism, there is high requirement of pyruvate. Talking about oxygen requirement, it is normal. In case of benign tumor, there is not much of the difference as compared to normal cells, whereas in case of malignancy, hypoxic conditions can promote the malignant progression. Hypoxic conditions can promote angiogenesis. Vascular endothelial growth factor release. The normal cells usually don't release such factors. In case of benign growths, there is again low or limited or regulated release of VEGF production, whereas in case of malignancy, there is higher and higher release of vascular endothelial growth factors. Talking about telomerase activity, the normal somatic cells do not have telomeric activity. In case of benign tumors, that this telomeric activity may start to re-express, but there is very lower extent of that, and it is still absent in most of the somatic cells in case of the benign tumors, whereas this telomerase is frequently reactivated in case of malignant tumors. This is the core reasons why we can say these malignant growths are immortal. Talking about local invasion, it is absent in benign tumors, whereas local invasion has to have occurred for the malignancy to happen. Local invasion is always present in case of malignant tumors. Even there is the formation of secondary tumors in most of the cases. Metastasis is absent in the benign tumors. Metastasis is present in the malignant tumor. The root may be either hematogenous, which means that the tumor is spreading through the blood vessels. It may be lymphatic, which means the tumor is spreading through the lymphatic system. There is transcoelomic dissemination, and sometimes there is neural dissemination as well. So let's talk about the characteristics of malignancy. What are the main characters of malignancy we studied until now? So there is genomic instability due to multiple progressive mutations. There is loss of cell-to-cell contact. There is loss of cell to extracellular matrix contact. There is number one, there is detachment of cell-to-cell contact proteins like cadherins. And second, there is decrease in expression of such proteins. And all this is achieved through dedifferentiation process as well, which we called epithelial mesenchymal transition. The cells will become self-sustainable. They will lose the dependence on mitogens to divide. They can divide even in the absence of any growth factor, any mitogen. There is increase in the release of lytic factors. These lytic factors will start degradation of basement membrane, and this degradation of basement membrane will allow the cells to invade the local tissues. There is dedifferentiation we talked about. There is loss of morphology. Cells will acquire some kind of oval, irregular type of shape. The contact with basement membrane has lost. The cells are no longer flattened at the basement membrane we talked about in our previous diagrams. There is increase of nuclear to cytoplasmic ratio, which means cytoplasm is decreasing, nuclear size is increasing. There is higher and higher expression of nuclear pods. The core reason is that the cell has to increase the processes of replication, transcription, translation, for which they need these nuclear pores to move substrates and all those proteins inside and outside of the nucleus. There is larger nucleolus, which is also required for these processes. There is Golgi dispersal. There is Golgi breakdown, Golgi dispersal. The cytoplasm looks pale in color. All these are the characteristics of the malignancy. Basophilic cytosol. The cytosol, which looks something bluish in color, we talked about this in the very first diagram right here. These cells, which are looking the dark purple, dark blue, these are the basophilic cytoplasmic cells. These are the significant tumor cells, and this happens due to increased production of ribosomal rRNA, and this increased production is to fulfill the translation requirements. There is significant telomerase reactivation. There is local invasion, there is distant invasion through various routes we talked about. There is formation of secondary tumors, there is colonization, there is angiogenesis, which is promoted by hypoxia, low oxygen, which promotes the hypoxia inducible factors, which will again stimulate the release of VEGF, and there will be angiogenesis, tumor growth and progression, ultimately leading to more and more spread of such tumors. Let's talk about the routes of the tumor dissemination or tumor invasion. The first route is direct invasion. We talked about this until now, that how the cells will degrade the basement membrane and invade the surrounding tissue. Let's move on to lymphatic route. Some tumors invade through lymphatic system. Many carcinomas show this kind of invasion, and therefore in such cases lymph nodes can act as important site to detect these metastatic diseases earlier. Then there is hematogenous route, where the tumor will take the blood supply, the capillaries, as the primary route of invasion. Both sarcomas and carcinomas does this kind of invasion. The target organs are almost sometimes lungs, liver, bone, and sometimes brain. The pattern will vary according to the type of tumor and type of mutations it has undergone. Then we have transcelomic or body cavity spread, in which tumors will utilize the peritoneal cavity or pleural cavity to spread itself. Tumor will detach from the primary site. They tend to take refuge in these cavities and spread along with time on these distant serosal surfaces. For peritoneal cavity, various associated cancers are ovarian cancers, the cancers of GIT. There are some abdominal malignancies, and for pleural cavity, there are certain thoracic malignancies as well. Let's move on to classification of cancer. On the basis of tissue of origin, we call it histogenic classification. There are various types of cancer. First one, if it is originated from epithelial cells, we call it carcinoma. And embryonically, these carcinomas may be either ectodermal or endodermal. So these are the carcinomas, which are epithelial cell cancers. Second one is mesenchymal cell cancers, which we call sarcomas, and embryonically they are always mesodermal. Then we have hematopoietic cancers. Examples, we have leukemia, lymphoma, plasma cell neoplasm. These are also a type of these mesenchymal cancers. These are also a type of sarcoma. Then we have melanocyte cancers. We call them melanoma. Then we have neural or glial cell cancers. We have germ cell cancers. We call germ cell tumors. Let's talk about epithelial malignancies or carcinomas. These are derived from the epithelial cells, and embryonically it may be ectodermal or endodermal in origin. So if it is derived from squamous cells, we call it squamous cell carcinoma, and these squamous cell carcinomas are mainly present in skin, oral cavity, esophagus or cervix. And second one is adenocarcinoma. If it is from the glandular or secretory epithelium, it is adenocarcinoma, and such kind of cancers are present in the digestive tract, GIT cancers like colon cancer, stomach cancer, pancreatic cancer, lung cancer, we have breast cancer, and we have prostate cancers, because all these organs are having these secretory or glandular epithelium. If we talk about other carcinomas, we have basal cell carcinoma, we have urothelial carcinoma, we have hepatocellular carcinoma, we have renal cell carcinoma, we have thyroid carcinoma. Let's talk about sarcomas, which are mesenchymal malignancies. Embryonically, they are mesodermal malignancies. If it is bone, we call it osteosarcoma. If it is cartilage, we call it chondrosarcoma. If it is muscle, muscles can be of different types. If it is skeletal muscle, we call it rhabdomyosarcoma. If it is smooth muscle, we call it leiomyosarcoma. It is adipose tissue, we call it liposarcoma. For fibrous connective tissue, we call it fibrosarcoma. For blood vessels themselves, we call angiosarcoma. Let's talk about hematopoietic lymphoid malignancies. Hematopoietic stem cells will give rise to two different types of cells. One is of lymphoid lineage and one is of myeloid lineage. And there are various cancers associated with these lineages. Talking about leukemia, it is the malignant proliferation of hematopoietic cells. There is two different types of lineage. It is either lymphoblastic leukemia or it can be myeloid leukemia. And these lymphoblastic and myeloid leukemias can be acute as well as chronic. Then we come to lymphomas, which is the malignancies of lymphoid cells. Commonly we call them solid masses. So there can be Hodgkin's, non-Hodgkin's lymphomas, and there may be involvement of B cells, T cells and natural killer cells as well. Last one is plasma cell neoplasms, which include multiple myelomas and plasmacytomas. Let's talk about neural malignancies or neuroectodermal malignancies to be specific. They are derived from neural or glial lineage. It has gliomas, it has peripheral nervous system tumors, it has neural precursor tumors. For gliomas, we have astrocytoma having involvement of astrocyte. Similarly, we have glioblastoma, we have oligodendrocytes in which we have oligodendroglioma, we have ependymoma. For peripheral nervous system, we have malignant peripheral nerve sheath tumor. We have neuroblastoma in case of neural precursor tumors. Let's talk about melanocytic malignancies, which involves the melanocytes. They are called melanoma. Melanomas are not carcinomas. They are classified as sarcoma-like distinct neuroectodermal malignancies because they originate from the pigment-producing melanocytes rather than the keratinocytes themselves, which are true epithelial cells. Talking about germ cell malignancies, if primordial germ cells are involved, we call them germ cell malignancies. Most common examples: seminomoma, dysgermimoma, embryonic carcinoma, yolk sac tumors, choriocarcinoma and teratoma. The important point here is this teratoma contains cells from ectodermal origin, from mesodermal origin and endodermal origin, which leads us to another important concept, which is mixed or multi-lineage malignancies, in which multiple lineages of tissues are involved. Examples: carcinosarcoma, we have teratocarcinoma as well. Let's talk about TNM classification of tumors. It is a standardized system for describing the anatomical extent of malignancy. So it is TNM. T determines the primary tumor, its size and extent of the local primary tumor. N concerns about the regional lymph node involvement. M is concerned about the distant metastasis, whether the cancer has spread to distant organs or not. There are different notations in each of these categories. For example, if it is TX, it means the primary tumor cannot be assessed. T0, which means there is no evidence of primary tumor. But we will focus is not TX or T0. We will focus is about this grading system, which is T1 to T4 grading. As we progress from T1 to T4, there is increasing or progressive size and extent of local invasion. For N assesses the regional lymph node involvement, and not, which means no regional lymph node metastasis has been seen. And again, we have grading, which is N1 to N3, which is increasing extent of regional lymph node involvement. Talking about M, which determines the distant metastasis. If it is M0, there is no evidence of distant metastasis. M1, there is distant metastasis, and similar grading will be carried out. Are given with an example of T3, N1, M0. What type of tumor is this? If it is T3, there is a relatively advanced progression in terms of primary tumor, because T determines the primary tumor, its size, its local extent. So it is relatively advanced primary tumor. If it is N1, it means there is a regional lymph node involvement. Involvement is not very much, but there is involvement started. M0, which means there is no distant metastasis seen. So we can say that the tumor is relatively local, but regional lymph nodes have been involved and there is no significant metastasis which is seen. And as an inference, we give a stage to the cancer. There is stage 0, there is stage 1, stage 2, stage 3 and stage 4. As we move from stage 0 to stage 4, the dedifferentiation will increase. Stage 0 means there is carcinoma in situ. There is no significant local or distant invasion. Stage 1 and 2 are generally localized tumor diseases. Stage 3, the disease has progressed more extensively, regional lymph nodes have been involved, and there may be significant involvement of lymph nodes as well. For stage 4, there is distant metastasis which is seen.